Hi folks, welcome to another short National 5 biology video. This time we're going to be addressing the topic of proteins and enzymes, which I know is one that students often struggle with, but hopefully after this you'll know the topic a little bit better. So let's kick off first of all by talking about proteins. Now there are 5 different types of proteins that you need to know about for your exam at National 5. Those are structural, enzymes, hormones, antibodies and receptors. Okay, now I know that's quite a lot to remember, that's quite a long list, so I've made a little way to memorise that, and that is what you're told from a very young age: always remember to share. Okay, if you remember to share, you remember S for structural, H for hormones, A for antibodies, R for receptors and E for enzymes. Okay, so if you remember to share, you'll remember the 5 different types of proteins that you need to know about for your exam. You also need to know what each of them does, what their function is within the body. So the structural one, as the name suggests, forms supporting frameworks and body structures. The enzymes, we're going to learn more about them in a moment, but they catalyse biological reactions. Hormones are chemical messengers. You'll learn more about them in Unit 2 if you haven't done that yet. And antibodies, also part of Unit 2, are part of the immune system, and they help to destroy pathogens by binding to specific ones. We'll talk more about them later. And receptors. Now receptors are little proteins in the cell membrane which bind to specific proteins. Okay, and again we'll talk more about receptors later on in the course. But you need to know those 5 proteins and their functions for the exam. So the key to remembering that, remember to share. Okay, now we're going to be focusing on this topic on one of these proteins in particular, and that is the enzymes. Now enzymes are biological catalysts, okay, and they are made by all living cells. Okay, you might have learned about catalysts in chemistry before. Catalyst is a substance which speeds up a reaction. Now a biological catalyst is simply something which is found in a living cell which speeds up a reaction. Okay, so they speed up cellular reactions and they're unchanged in the process, just like any catalyst. Again, you might have studied this in chemistry if you also do that. Now the shape of the active site, okay, that's this area here on the enzyme. Okay, the shape of that active site is complementary to a specific substrate or substrates. Okay, so it's complementary to a specific substrate. Now we've used that word complementary before when we were talking about base pairs, which means that only one particular thing goes with another particular thing. In this case, only one substrate fits in or is complementary to one active site on an enzyme. Okay, and if we have a look through how an enzyme controlled reaction actually occurs, we have a look at stage 1 here. We've got our enzyme, okay, we've got our active site here, okay, the shape of the active site there you can see is complementary to the shape of our substrate here, okay, our little red substrate which will move into and bind to the active site. Okay, so the substrate binds to the active site, step 1. Step 2, over here we have the substrate bound to the active site, and this forms what's known as the enzyme substrate complex. And this is when the reaction will start. Okay, the reaction will start to take place once we form the enzyme substrate complex. And then at the end, after the reaction has taken place, there are now products, okay, which have been made as a result of the reaction, and those products will be released from the active site, and the reaction is now over. The enzyme controlled reaction is now over. So there are 2 types of reactions we need to know about for the National 5 Biology course. They are called synthesis and degradation. Okay, synthesis is where we build up smaller molecules into larger molecules, as so. Okay, both of these are examples of how it might be displayed in your exam. Okay, smaller molecules into larger molecules. Degradation is where we break down larger molecules into smaller molecules, like so. Okay, so substrate on the left, product on the right. Large molecules to small molecules. Synthesis, small molecules to large molecules. You need to know the difference between those 2 types of reactions, as well as being able to identify them from an image. Now the factors that affect enzyme activity, there are 2 main ones that we need to know about for the exam. The first one is temperature and the second one is pH. Okay, and they both have a an effect on enzyme activity depending on the particular temperature or pH that the reaction's taking place at. So for example, for temperature, when we have low temperatures, the enzyme activity is relatively low, but as we increase that temperature, slowly the activity lifts and lifts and lifts till it reaches its maximum point. Okay, the maximum enzyme activity is called the optimum. Okay, we call it the optimum, that's a key word to remember, where the enzyme activity is at its maximum. But then after that optimum, you notice there's a big drop off in enzyme activity very quickly. And that's because we've reached a temperature at which the enzyme becomes denatured. Now, when an enzyme becomes denatured, its active site changes from this normal shape, which fits well with the substrate, to this misshapen active site where the substrate is no longer able to bind. Okay, so we see that the enzyme's active site has changed shape and the substrate is no longer able to bind. Okay, and it's a similar idea over here with pH. At a very low acidic pHs or very high alkaline pHs, we get denaturing of the enzyme. Okay, at both low and high pH, we get this denaturing. But right in the middle, we've got a sweet spot. Okay, the ideal pH, the optimum pH, where the enzyme activity is at its highest. Okay, so the two factors we need to remember, temperature and pH. When they're both at their maximum, we call that the optimum, the maximum enzyme activity. And then after a certain temperature, it will denature, or at extremely high or low pHs, we'll get a denaturing of the enzyme, and that's where the active site changes shape. Okay folks, I hope that was helpful and you've learned a little bit more about enzymes over the course of this short lesson. Let me know in the comments if you've got anything you're still not sure about or if you enjoyed that lesson, and I'll see you in the next one. Class dismissed.